Good afternoon, everyone. Welcome to the Mycology lab of BIOL1263. In this lab, like we discussed in class, we're going to go through the morphological and reproductive structures of two different classes of fungi and two different types. I just want to run through a brief introduction with you all, right? So remember, like we discussed in class, fungi are a class of eukaryotic organisms, or type of eukaryotic organism, right? And fungi could be found on fungi. All fungi are saprophytes in that they get their nutrition through absorptive nutrition. So what you're observing now is the cleothecium of some ascomycetes growing on the roots of a dead plant, right? And if I move the sample around, you can see the hyphae of that ascomycota species of fungi germinating out of the cleothecium, right? These ball-like structures, remember, are the fruiting bodies of some ascomycetes, and they contain inside them the spores of that ascomycete, right, or the ascospores of that ascomycete fungi, right? In today's lab, you're going to look at three different types of fungi, right? And the three types of fungi include the penicillium fungi, which is an ascomycete, right? Remember the penicillium, remember the ascomycete, they produce ascospores, which are finger-like projections, right? And we're going to pull that up on the microscope for you in a second. The other two types of fungi include rhizopus. Include rhizopus, right? And this is what it looks like on a petri dish, right? So the petri dish contains nutrients in a solid medium that we place a part of the mycelia onto, and then that grows through elongation of the tip. What you're observing on the petri dish is the mycelia of the fungi, and you can't look at a single hyphae, but that mat of threads growing on the fungal plate is what we term the mycelia of the fungi, right? If you look closely on this fungi, you could see some dark ball-like structures growing along. So the white is the mycelia, but the black spots along the mycelia is the fruiting bodies, and the fruiting bodies is what is the asexual reproductive structures, right? So it's very unlikely that you'll see sexual reproductive structures grown on a petri dish, but it's more likely that we see the asexual fungal bodies, right? So the zygospores, the sporangiospores, and all of that. Prepare the microscopic slide, we put a small sample of the fungal mycelia from the petri dish onto a slide, right? There's no need to stain these fungal mycelia because through the light microscope, through the light microscope, right? Light is able, these are more or less transparent, and light is able to pass through them without the need for a stain, right? Go up one. So on the picture of the slide from the microscope, you can clearly see that this is a zygomycete. And what you're noticing is that these dark structures here are the zygospores, the sporangiospores, right? Surrounded by the sac, right? The entire structure is called the sporangiophore, right? And the hyphae leading towards the fruiting body is what provides the spores, right? So spores bud off the hyphae and collect in this sac, right? And then they are clones of the mother hyphae, and they are asexual spores, right? Note that in this zygomycete, the hyphae is aseptate, so there's no sections between the hyphae. So you want to make notes like that, right? So in your observations, you're going to draw the 3 types of fungi listed on the lab handout, right? You're going to look for Rhizopus, Penicillium, and Aspergillus, right? You're going to draw the asexual fruiting body as well as the hyphae type in it. If you come across diagrams of the sexual spores, right, then please make note of that. And in your drawings, make note of all the different sections of the asexual and sexual fruiting bodies.